Hi everyone, I'm Tom Osborne from Blacksheep Sciences, and I'm really pleased to be kicking off our new Droplets and Deals series. And for the first episode, I've got Chris Tame here from Ternary Therapeutics. Thanks for joining us, Chris.
Hi Tom, yeah, thank you very much for the introduction.
No problem. So to kick off, what is one thing that people might not know or expect to know about you and your journey so far as a scientist and as CEO of Ternary Therapeutics?
Yeah, so I actually started my career straight out of university as an undergraduate. So it's quite traditional for, or typical for scientific founders to have PhDs and extensive postdoc experience, but actually there's more than one way to grow your career. And I actually started at GSK as an undergraduate and learned on the job, and then later did my PhD in collaboration between GSK and the University of Strathclyde.
I think you were actually if not the first, one of the first batch of industry placement PhDs out of GSK. Is that right?
That's right. Yeah, I was. There were a few years in front of me, but yeah, it was definitely an opportunity for both the institutions and the students to learn as they went on. And now it's an incredibly successful scheme with hundreds of students that have graduated both through chemistry, biology, and DMPK sciences at GSK in Strathclyde.
And I think actually you were saying the other day that you were going back, is it 20 years since, or maybe it's not that long, but there's some sort of anniversary recently that you were talking at and encouraging the PhD students to, kick on their careers.
Yeah, my first interaction with GSK was actually back in 2005, up to 20 years ago, where I had an opportunity of doing a week-long placement at GSK to get an opportunity to understand more about the pharmaceutical industry. And it's a scheme that is specifically set up for students that go to universities that don't have extensive connections with the pharmaceutical industry, and also for first generation undergraduates. So that was the first time I had the opportunity of being at GSK and getting that flavour for what a career in pharmaceutical research would be like. That scheme has obviously now had its 20-year anniversary, and there have been 700 students that have been through that course from all over the world. So it's an incredibly successful scheme that gives a lot of people access to this type of career that otherwise would find it difficult to do so.
So for those who don't know, can you give us the short version of what Ternary Therapeutics does?
Sure. So in a nutshell, we use AI to design a type of medicine known as a molecular glue. These are small molecules, and they work by gluing two different proteins together. So these are natural biological processes that are disrupted in disease, and we correct that biology by gluing the proteins back together again. You're muted again. We'll get there.
Sorry, I'm going with my script. So what's the big problem that you're solving? Like, are there other people doing molecular glues? Like, what really sets you apart, and why does it matter right now?
So 90% of genetic drivers of disease are actually caused by inactivating mutations. So these are changes in the proteins that stop its function. But actually only a handful of drugs work by reactivating those proteins. Most of the drugs that we take block the action of drugs. What we do is we use molecular glues to reactivate proteins and restore natural biology. So this is fundamentally a different approach to not just molecular glues, but all types of therapeutics, and it gives us that unique opportunity of actually directly targeting the genetic drivers of disease.
That's really cool. So you're one of the only companies that's doing this, making proteins work again, as opposed to stopping proteins from doing bad things.
I think there's definitely ways that we can maybe upregulate disease expression of proteins to try to control it, but notoriously that's quite difficult to control, and you can overactivate those proteins, and that can lead to safety issues. But typically molecular glues have been worked as in a degrader setting, and other therapeutics work by inhibiting or blocking the action, whereas we're trying to actually restore the biology by gluing those existing proteins back together again. So it's fundamentally a much safer and much more targeted approach.
So let's talk a little bit about you and your role. Like I think we mentioned earlier, you're co-founder of Ternary Therapeutics, and this is your first time as CEO. So what was the most surprising thing about stepping into that role as CEO?
The thing that really surprised me was how many people in my network were willing to give me a lot of their time to help me make that transition. People in my career that I've just met and worked with in a scientific context, and never really expected to make those connections for a specific reason. And actually, when I was making that transition, how much time they took and helped introduce me to other people in their networks to help understand and try to learn from some of their lessons from building companies. That's really made the difference for me, I think.
Sorry, and in this series, we'll talk to a lot of people about who've making transitions from a science background into a more commercial background or management role. So which of those skills from your science background have helped, and which sort of didn't translate at all and you had to kind of suppress your natural tendencies?
I would say one of the things about core scientific disciplines is that they're highly translational, and actually thinking analytically and planning things out methodically and testing hypotheses is all the same skills that you need in business as well as in science. It's difficult to think about areas of science that are hard. I think really deciding when to stop working in areas. I think we can all as scientists think about the next experiment we would do, but there has to become a time where you actually reach that no-go decision, you stop and you move on. And I think that's the hardest transition when you're moving from a more academic mindset to a commercial mindset.
Yeah, I agree. You always want to find out the next thing. It's always, as a scientist, the anomalies are the interesting things when you're doing scientific, sort of pure scientific experimentation. And then when you move into more commercial-faced roles, you have to sort of suppress those and ignore those and be like, know when to stop and stop looking at those interesting little intricacies. So if you had advice, or if another scientist asked you for advice about becoming a CEO or founding a company, what would you tell them, or what advice would you give them?
So I think that the CEO role that I currently have is, I'm enjoying myself more than I've enjoyed myself my whole career. It's incredibly fulfilling. It's great to come together with a team of people, create a vision and a culture and execute. So therefore, I think it is, if you want to become a CEO, I think that you can't do it all on your own. You need a strong team around you. You need people that you can trust. So I think it's really important to be able to build that. It's not a glamorous role. Like, I spend most of my time thinking about all the ways things can go wrong and think about how we would mitigate those risks or approach them. And when I'm not thinking about how things can go wrong, you have to deal with rejection, because on a weekly basis, fundraising is a tough environment to be in. So you have to have a lot of resilience to sort of deal with those risks and those things while still motivating the team and getting excited and being able to actually work with the team on progressing internally. So it's a challenging role, but it's incredibly rewarding.
Yeah, you mentioned about sort of fundraising there, and we talked about moving from science to CEOs, and there's different sort of types of CEO as well. Myself, I'm a different type of CEO to you, in that I think I'm much more operational and trying to make things happen. I'm working in a sort of revenue generating company, whereas I think your role right now, at least, is very much fundraising and raising money. Was that a particularly hard jump to go from science to raising money as a company, rather than sort of operational science management?
I think when we get down to it, it's no different to writing a grant application as an academic, or trying to convince your boss that there's a particular area of work you want to work in. Ultimately, you're just convincing a different person. You have a good idea and you need to convince them to give you some money to go away and do it. And there's going to be different upsides, and you have to understand the people that you're asking for permission and support to do that work, and you need to make sure that your messaging is clear and concise and they can understand the benefits of doing it. So fundamentally, I don't think it's a very different ask to transition from, say, a science role where you're seeking, let's say, stakeholder approval within a large company or a grant body for money to pitching to a VC for fundraising. I think the biggest challenge, though, as a scientist, when you're trying to pitch to VCs is making sure you can never make your message simple enough. You need to have a lot of discipline about distilling down all those technical details that you think are really important to the message to really understand what are the things that really matter. It needs to be very clear exactly what you do and why you're different.
And was that the hardest bit about explaining about fundraising, was explaining your research to investors?
I think in general, VCs are very easy to communicate to, and they are a lot smarter than maybe what some people give them credit for. Especially if they're going to invest in an area, they spend a lot of time making sure they're experts in that space. I just think it's about being clear, being clear with your messaging, and understanding which bits are the most important bits to communicate. And I think it's the challenge around dealing with rejection. It's not personal. Different people have different approaches, different theses, and it's just trying to match the individual company with the investor to make sure that both of those visions align. Obviously, I've just mentioned about some of the things that I felt about transitioning from science to CEO, and as you said, you've also made that change quite recently. So, does anything that I say resonate with you, or do you have a different perspective?
Absolutely. I think I don't think about it too much. I still feel like a scientist, and when I transitioned into more commercial roles, I transitioned in commercial roles because I wanted to stay involved in really cutting edge science and cutting edge things, which when you're at the bench, you can get sidelined and sidetracked into doing what you're told to do and into sort of very niche areas of research. And so transitioning to a commercial role allows you to do cool stuff and stay in that cool stuff and stay at that cutting edge of science. And that's what I really like about it. And I kind of still feel like a scientist. So my scientist would tell me I'm not, because, because I tell them, stop that, don't do that, that's a waste of time, because we're trying to focus on getting something that's commercially viable or is important and is going to make money. And that's ultimately that's the biggest change, is that what you're focused on trying to do is make money and not make publications. You still have to have that scientific rigor around what you're doing, but the end product of what you're doing is a pound or a dollar sign and not a piece of paper.
Yeah, I think also you can do both. I think we published just a couple of months ago, and like you say, there needs to be. You need to think about what the ultimate goal is, and ultimately, if in my industry we're thinking about a therapeutic asset, a medicine for a patient, and if we are genuinely providing a medicine that will give the patient benefit to something existing, then there will be commercial value in that, and we just need to make sure that we're really laser focused on that. But at the same time, we want to be very innovative about how we develop those, and with that comes science that we hope that we can share with the community and the community can learn from, and that's why you can balance that need of contributing to the scientific community whilst also thinking about developing commercially viable assets.
Yeah, absolutely, I agree. And everything that we do, almost everything that we do at Blacksheep Sciences has some kind of, there's a tangible benefit to people in general. And most people don't go into life sciences, whether that's drug development or tool research or anything like that, without thinking that what they're going to do is going to make a difference to people. And so then if we sort of go back to that thing about being a CEO, and it's do you ever get, because I don't yet think I've cracked it. Like I know exactly what this is all about. Like, do you think you've cracked it? Do you think like, yes, I'm bona fide CEO material now, and I'm just going to crack on?
Absolutely not. No, I don't know if you ever will. Um, One of my early mentors always said to me, Chris, the day you stop learning is the day you stop being useful to me. And I sort of like take that as like a philosophy throughout my whole life. I don't think that you can really, you're ever the finished article, and maybe life would get boring if you were. I'm still very early on in my journey as a CEO. I'm still learning a lot and really enjoying that journey. I think that every challenge, like I think that being a CEO now in biotech has unique challenges to say 5 years ago in terms of the sort of macroeconomic environment, fundraising environment, how to build companies, and I think that the environment that you're working within is always changing. So I think there's always going to be like unique challenges that you have to adapt to and learnings.
Yeah, one of those unique challenges, or actually, it's not a unique challenge, but it's a new challenge for any company is AI. How to make the most of AI. Our company Black Sheep and what we do, we don't use a lot of AI tools. We do a lot of mechanical engineering and pure scientific research, and we're trying to use some of those AI tools in that. But your company, Ternary Therapeutics, that I believe AI is sort of a core fundamental component of your workflow. So can you explain a little bit about, how you're using AI and sort of how important it is to you?
Yeah, sure. So one of the challenges when you're trying to design molecular glues is understanding what the ternary complex looks like, the structure of the ternary complex. So that is how the molecular glue sits between two proteins and glues them together. And traditional technologies, experimental techniques like crystallography and cryo-EM are quite challenging to adapt to this unique challenge of two proteins working together, interacting together. And you also have additional challenges in terms of being able to isolate and work with these proteins. Some of them are just too large and flexible to study in this way. So as we know from like the 2024 Nobel Prize in Chemistry was awarded to a group of researchers that invented protein folding AI. So this is a type of structural biology that can take a linear sequence of amino acids and use AI to fold it into a 3D protein structure. And it's absolutely transformed our understanding of protein structure and function. We're seeing lots of applications. So this is an area that we're focusing on as well, is structure biology and how we can use computers to simulate the ternary complexes that the molecular glues occupy, and then use that to design better molecules. So we have like an end-to-end platform that looks at whether a target can be gluable, and then what the ternary complex looks like, and then finally how to design molecular glues using that. And each one uses different types of AI involved. And as I said, the reason why we're doing that is because the experimental approaches simply aren't fit for purpose.
So your workflow is using various computational approaches to predict the success of a molecular glue in what it's going to do. What are some of how do you validate that in the lab? Like, is that something that you're also working on?
Yeah, and it does actually require some sort of innovative approaches in terms of experimental approaches. So we're using a technology called limited proteolysis mass spectrometry, which is a way to detect these ternary complexes and protein-protein interactions in cells so that we can actually study them in their proper environments. And one of the things that this allows us to do is to understand where and how these proteins are interacting with one another. Now, that information on its own isn't particularly useful. You can't use that for design, but actually that's the perfect level of data that the AI models need to then be able to actually elucidate a really highly resolved ternary complex structure. And obviously, the proof of the pudding is always in the experimental evidence and the quality of that data. So we still have to rely, obviously, on a lot of very good synthetic organic chemistry to develop the molecular glues, and then also to develop the cellular and protein assays to fully characterize the drugs.
So is that really the limitation in the workflow, is the amount of experimental data that currently exists? And so if you can create a lot more experimental data, does that make your AI models that much more robust?
Yeah, I think one of the big limitations of AI models is that they like to predict things that they've seen before, and therefore, if you're trying to use it to predict something that hasn't existed, then it's not very accurate at doing so. And that's why I think that actually you need to be able to use AI in combination with an imaginative scientist, and to be able to use it in tandem. But the way that we actually specifically use AI is we use it in a transfer model approach. So we actually use data sets which are in abundance, single protein data sets, protein ligand interaction data sets. We train models and then we apply them to molecular glues. And in that way, we don't need to generate lots of data on ternary complexes to train the models. I think it's also fair to say that we use a suite of computational approaches. Not all of them use AI. So we also employ a lot of physics-based models, molecular dynamics, and I think that actually the balance and the sort of synergy between machine learning and physics-based models give you much higher accuracy than if you just use one of them alone.
I think you said something there that resonated, and you said it to me before personally. I can't remember if it was dropping the kids off at school or playing cricket, but was that the sort of the major limitation of AI is that inventive step, and that's, I think I still hold dear to that because I think it's what keeps us at work and keeps us, interested in science. Because otherwise, if we just hand it all over to a computer, then what's the point in us being here?
Yeah, well, I think it's smart scientists developing smart AI, and I think that the level of data that we have available to us and we're able to generate now is too big for a human brain to comprehend on its own. So using computers to reason through that information and help guide us is really useful. I think one of the fundamental limitations of AI in biology is just biology is challenging to reproduce, and understanding what data is trustworthy, reproducible, and not is really difficult. And that's before we even think about the translational challenges between preclinical work, where we may do studies in a test tube, to then putting it in a human, and that translational effect is a really significant challenge. So I'm not saying that AI won't provide a solution, it will, but it'll only do it in combination with a smart scientist. And at the moment, in science, we're not at the stage of driverless cars. We aren't able to identify all those variables and generate data for them to train a model on them.
And I think if we understood it all, it would become boring. Well, Chris, thanks for the chat, and thanks to everyone for listening.